Um, Hello, everyone. Good evening who are joining from India, and good morning who is joining from United States. So my name is Parth Desai. Dr. Gopal, thank you so much for this kind introduction, and I'm feeling very honored and humbled to be on this platform today. So thank you for giving me this opportunity. So my topic is SiRNA and ShRNA mediated gene regulation. Everybody can hear, right? Just want to make sure.
Yes, you are audible, very well audible.
Wonderful.
Yes,
So today's topic is SiRNA and shRNA mediated gene regulation. So now there is a little difference in gene regulation versus gene manipulation. So gene regulation where we regulate the functionality of the gene or its outcome, and the gene manipulation is something where we manipulate the genes. That means we can edit them. But today's topic is on gene regulation. So agenda for the topic is introduction. I will walk you through the introduction, then mechanism of gene regulation, RNAi mechanism, shRNA mediated silencing, comparison of siRNA and shRNA, applications and therapeutic potential of this uh, RNAi or siRNA and shRNA, and then experimental procedure. So introduction, what is gene regulation? The process of production, functionality, and the expression of genes, right? So everybody knows that genes are being expressed through transcription. But what happens after that? That comes translation. So gene is functional and it is performing its functioning, and genes are regulated in different ways. But we don't know. I still remember my second internship that was in ISER Bhopal. I wrote a nice email to a professor asking that, I know that you are working on epigenetics, but I have this simple question that how these genes are regulated automatically inside the cell or in our body, because they do not have brain to function. There is nothing to give them a signal. Still, they are continuously working. That simple curiosity triggered me into the world of epigenetics, gene regulation, and furthermore. But yeah, so the purpose of this gene regulation is to control the RNA functionality and the gene functions. The importance of gene regulation is it is crucial for cellular processes like cell differentiation, energy production, and homeostasis. So we can say that this gene regulation is actually the quality control and quality assurance department of a cell, because gene is transcribed, it is monitored, and it is regulated in a very in a very well manner to to give every cell a routine functional or functionality. But when these genes regulation got disturbed, it turned into the disease like cancer and so many others. So that's why the gene regulation is very important. Introduction to the mechanism of gene regulation. So there are different gene regulation. The mechanisms are transcriptional gene regulation, post-transcriptional, translational, post-translational, and epigenetic. So these are the different stages or the levels where gene regulation occurs inside the cell. As I said, it is the quality control and the quality assurance department of a cell. That genes should function very well. Now, what is the transcriptional control? Gene regulation. How much mRNA is being produced? That is one thing. Second, in the post-transcriptional mechanism of gene regulation, what happens? So once the RNA is produced, now the mRNA processed via alternative splicing and mRNA goes to the stability, because the stable mRNA functionality is very important to produce the protein. Now, translational. How much mRNA is processed via the tRNA, ribosomes, and other translational processes. So the transcription happens and it controls that how much mRNA is produced. Now, after the mRNA is produced, it is being stabilized, and then alternative splicing happen to make sure that what type of protein it's going to produce. And then how much mRNA is processed via the ribosome, tRNA, and other translational process. That is the translational mechanism where the gene regulation happen. And the post-translational, where the protein can be activated after the mRNA is being translated, modified or degraded via phosphorylation, ubiquitinylation, and proteolytic cleavages. And epigenetics, epi is around. So that's what I learned in the scientific field, that every time if we break it down, we know where these things are happening. So epigenetics is around the gene or outside of the environment of the gene, where at the DNA level or the histone level. So changes to DNA and histone that includes the modification like methylation, acylation, and others. This is all different types of mechanism of the gene regulation. Now, moving forward, RNA interference. So when it comes to the gene regulation, today we're going to talk about siRNA, which is silencing a short interfering RNA or silencing RNA, and the shRNA, which is short hairpin RNA. So RNA interference is the mechanism through which this both gene regulation occurs and happens. So let me walk you through this step of RNA interference. The very first step is the production of double-stranded RNA, which is dsRNA, and the exogenous RNA inside the cell. So once the RNA is being produced, then the second step is this particular double-stranded RNA is recognized by the dicer, which process the dicer into the small interfering RNAs or siRNAs, which is 21 to 23 nucleotide size only. Now, once the siRNA is made, it goes through the RISC complex, which is RNA-induced silencing complex with other proteins, where the passenger strand of the double-stranded RNA or the siRNA is degraded and the guide strand will continue forward. Step 4 is siRNA strand combined with the RISC complex and target the mRNA. Step 5, the argonate protein recognizes the mRNA strand and cleaves the mRNA. Now, the mRNA is cleaved. What does that mean? mRNA will be degraded and protein will not be produced. So this is how the RNA interference works. Moving on, the second mechanism is ShRNA mediated gene silencing, where these shRNA are delivered in form of DNA vectors. And I will cover the comparison of how the siRNA and shRNA works. But once it is delivered inside the cell in the form of DNA vector or the plasmid, and once it is transcribed, it will form a stem loop. A stem loop is an extra loop where I can show you in this figure. If you see this is the stem loop. And the shRNA is produced by the enzyme called DROSHA, which will make the precursor shRNA and transport it to the cytoplasm. So as we know that this everything happens inside the nucleus, and the shRNA has to come out of the nucleus to the cytoplasm. In the cytoplasm, it will further process by the dicer into siRNA. So shRNA, after processing, goes to the siRNA only. And this mechanism I already explained you for previously. The siRNA-like molecule will be further incorporated into the RISC complex and attached to the mRNA target sequence. Step 5 is the RISC guided by the siRNA binds to the target mRNA and degraded. So that's how siRNA and shRNA works, but there is a little difference in both of these. As their names are different, their functions are, their production is also different. But the function is same, to regulate the gene or the protein production. So here is we see that this is the siRNA. siRNA short in 21 to 23 nucleotides. They are processed from double-stranded RNA by dicer. siRNA can be delivered using nanoparticles, ribonucleoproteins, RNPs, and other delivery methods. So siRNA is delivered in different methods. And they can be synthesized also outside of the cell. And siRNA are used for the short-term gene silencing. Their general turnaround time is 3 days max, or maybe 7 days is the maximum. But I never seen in my personal experience, I have seen maximum 3 days that I used to terminate my experiment after introducing the siRNA into the cell. Now, this is the siRNA with this extra stem loop. Sorry, this is the extra hairpin loop. ShRNA is longer that form the hairpin structure. So as you see that siRNA is this single double-stranded. This is the shRNA where it has an extra loop. shRNA are processed to become siRNA. So this is the difference as we see that. shRNA are delivered in a DNA vector form using the viral and non-viral transfection, where we see that siRNA is delivered using nanoparticles, ribonucleoproteins, and other delivery methods. shRNA are used for the long-term gene silencing by integrating into the genome. So shRNA goes through the DNA vector. They goes inside. They can be transiently expressed or they can be incorporated into the genome of the cell using viral based delivery. But once shRNA is incorporated, what happens is it will be continuously transcribed. It will produce more shRNA. It will go through the similar process that I explained previously, become an siRNA, target the mRNA, and degrade the protein. But this effect is very long term because they are now integrated inside the genome. And that's how they keep producing shRNA, and the cells will, and it will keep degrading the protein. So that's how shRNA regulation works differently than siRNA. Applications. We always think that, okay, we have this molecular tool or the technology. How are we going to use it? What are the different applications of this? So starting with siRNA application, it is used for transient gene knockdown in sensitive cell types, where some of the cells are not targetable with the viral or DNA vectors, because DNA vectors takes a lot of energy of a cell. Imagine a very sensitive cell type, right, which is very naive, like a small child or very sensitive. You cannot use the same things for the small child as we use for the adults. So you need different things. The same with the some of the sensitive cell types where the siRNA gene knockdown is helpful over the shRNA. siRNA is explored for the therapeutic purposes, specifically for viral and cancer-related therapeutics. siRNA can also be used for screening purposes, understanding the gene function and potential drug target. And I'll tell you, we are living in this world where gene editing or gene manipulation by CRISPR technology is very famous. I have seen a lot of graduate students, or including me, like I was so excited to use CRISPR in my experiments. And I thought like, okay, I'm gonna delete this gene and see the functionality and stuff. But we scientists have to understand that we are hardworking, definitely, but sometimes we have to find the easiest way to understand the protein functionality or the gene function. In order to do that, the easiest way over the CRISPR is siRNA, because siRNA just delivered easily inside the cell. It can be taken up. siRNA comes with lot of modification on the tail so that while you deliver, it will not be degraded by the other enzymes inside the cell. And I was amazed to learn about those different modifications, but it has a separate different topic. What I'm focusing on is siRNA is the easiest technology to start with to understand the protein function inside the cell before we move to CRISPR. Application of shRNA. shRNA are used for long-term gene silencing introduced inside the cells to see the gene knockdown over several generations. As I mentioned, once it is incorporated inside the genome, it will keep producing shRNA and it will keep targeting the protein. So that's why over several generations, the cells will not have that protein. shRNA used for the cells, those are difficult to transfect with siRNA. So some of the cells which are heavy and they do not have easy cell membrane, or they have a very different type of characteristics, then shRNA is used because we can use heavy viral load to deliver this type of shRNA DNA vector inside the cell. And because those cells has a receptor like HEK293, I know that their cells express very good viral receptor so that it just get engaged and lentivirus can introduce the shRNA plasmid. Or talking about macrophages, where adeno associated viruses, there are different type of virus we can use, but you can use adeno associated viruses combined with the shRNA vector, and it will deliver the gene of your interest or shRNA vector inside the cell. So those cells are easy to work with viral load. So definitely we can go for the application with those type of cells in the shRNA plasmid. ShRNA can be designed with inducible promoters that give the control over the time of its expression. So it's all about transcribe. That's why it has to be near the promoter or after the promoter. So we have to design the shRNA separately. Now, what is the therapeutic potential? Because we talk about gene regulation, it is useful for screening the protein function inside the cell and lot other things. But now researchers are moving towards how we can make this therapeutic potential of SiRNA, how we can use this SiRNA for therapeutic potential. So I came across these two articles where one of the article talks about different delivery therapeutic targets for the cancer therapy particularly, and another, the small interfering RNA based therapeutic application against the viruses. If you read these through, this both the articles, I do not have time to go through the details, but it is very informative. While I was going through it, I discovered that there are so many clinical trials right now going on with the siRNA in the particular cancer therapy. I find out that the very first siRNA based therapeutic was FDA approved for one of the diseases. I don't remember the name, but it was in 2018. So 2018, the very first FDA approved siRNA therapeutic came out. And after that, there are tons of clinical trials going on, which is written in this particular review. Definitely recommending to get more knowledge. Same with the viral for the viruses and therapeutic application of siRNA. So please go through this. You can take a screenshot or write this down. Now, moving forward, the therapeutic potential of shRNA. Now, it's a short hairpin RNA. Now we know that. CLED, in collaboration with the Horizon, got involved in developing smart vector to employ the shRNA-based therapeutics using CAR T cell therapy. Can you imagine like how the researchers have moved forward? The CLED is one of the companies. So you can go to cled.com and read more about the shRNA-based platform that they are developing to make the CAR T cell therapy more effective, because there are a lot of side effects associated with CAR T therapy and what they are trying to mitigate using the shRNA. An approach has been made to alleviate the chronic anxiety and age-related cognitive decline using the shRNA by targeting HTRA2 gene on 5-HT2A receptor. This particular title of intranasal delivery of shRNA to knock down the 5-HT2A receptor is really recommended read to understand what is the therapeutic potential of shRNA. This is not yet in the clinic. It is still in the early discovery part, but still it it provoke our mind that how the research is moving forward with siRNA and shRNA, and definitely needs to be considered the clide.com, where the company has talk about everything in what is their platform and how they are developing. So now we know now RNA interference, we know silencing of RNA, we know shRNA. Now let's talk about the workflow of experimental setup. We all are scientists. Some of you are, I know that might be doing it for years. Some of the students are joining today. So my whole purpose to deliver you the real experimental setup, that how we can perform SiRNA and shRNA into the laboratory with very effectiveness and very efficiently, so that we know what is the protein, what protein is doing. And if somebody asks me, if somebody asks a question that, okay, how did you do this and why did you do this, because the why is very important in research. If you want to identify a gene or a target, you need to know what protein it produces, right? So to produce that protein, and we need to understand what that protein is lethal to the cell or not. Because I have seen sometimes if you target siRNA and protein gets degraded, the cells will die because it is the most crucial protein for that cell to survive. But that can lead to a lot of new drug discovery. In cancer, we know that we are targeting cancer cells only, but the chemo targets all type of different cells. But imagine if we can identify those simple proteins that is produced by cancer cells, and if the cancer cell will die, that could be the new protein target. So that's why these techniques are important in drug discovery as well. So first of all, identify your target gene. That is the very first thing. You need to identify which gene you need to target and why, what protein it produces. Then go to the NCBI and show the FASTA format. So I would like to show you a little demo for the time being, and then I will show you what is APE plasmid editor also. So these three things I go through in my personal experience and experimental procedure. When I target any protein, I make sure that I have a sequence of the protein. I make sure that what are the criterias and everything. So quickly, I will get out of the presentation and I will show you the, let's say, just write down HPRT gene sequence NCBI. So this way, we will go to this particular website. Everybody can see this, right? Just one of you can say yes.
Yes, sir.
Yes.
Okay, good. So as you see this now, that we are on this website, I just simply write down that HPRT gene sequence and NCBI because these browsers take keywords. And it's so easy right here. I'm at my database. I'm at the sequence. Now in the sequence, I can go through the summary of this gene. I can learn more about the gene, genomic context, genomic region, transcript products, expression, bibliography, phenotypes, variation. So we can do a lot on this particular browser or portal. But my main important thing is here, genomic transcript region and products. When you go here, click on the FASTA file. When you click the FASTA file, you get this big sequence. Now this is the DNA sequence, right? What I do is I copy this particular thing by control C. I need to select. I will select this whole region like this, and I will copy this. Simple. And this is the APE, a plasmid editor. I use this. I have personally downloaded, and it is free. Okay, we do not have to pay anything for this service, and it gives amazing functionality to read the gene sequences, to compare the sequences. Even after you do gene editing, you can compare the after gene editing and before gene editing sequences. This is very good plasmid editor. There is a tutorial available, so please go through this and write down this. So what I will do is I can show you the plasmid editor that I have here. So I have a shortcut on my desktop. If you see, I will just copy and paste. And as we see here, now just find the ORFs, find the next, and you will see the find next ORS. Wherever you will click the cursor, it will show you the ORF. These features help me to identify. There are different features. This helps me to identify the open reading frame, then the start codons. Where is the start codon here? So that I can target my sequence. Okay. So I hope this is understand. Everybody can understand this one. Now coming back to the slide. So what I explain is identify your gene, download the FASTA from the NCBI. I can download or you can copy and paste, and then go to the APE plasmid editor where you will find out the ORF, the start codon and everything. Now you might be asking why the start codon, right? Because there are certain criteria. I will show you those criteria also. How you can target your sequence or design your siRNA and everything. Identify the start codon at the beginning and the end of the sequence. So whatever sequence you are targeting or the gene you are targeting, you have the whole sequence now, and you can identify which region you would like to target, because one siRNA will not work. So you need to use multiple siRNA to target the same gene to identify which one has the best potential to degrade the mRNA or to stop the function or whatsoever or downregulate the gene. Then design the sequence or order it pre-made from the library. So I will give you some resources where you can go through the company website, you can learn, you can educate yourself what are the criteria of siRNA and how these companies are working, and you can ask them questions if you want to order pre-made. Now, after this, we need to, after now we order the siRNA is with us, we can modify the siRNA with the fluorophores, with the some of the protection and that will not degrade the siRNA, the enzymatic protection. Like any, there are different type of modification available for siRNA, which will help siRNA live long inside the cell once it is delivered and not being chewed up by the enzymes. After we have to deliver now siRNA or shRNA plasmid, there are different delivery platforms. Either you can do electroporation, if you know your cells are capable enough to to bear the electrophoretic voltages, or the simplest way that people use is nanoparticles. There are different type of reagents available which says, okay, this is RNAi max, that's one I know from Thermo Fisher, but there are multiple. You can pick according to your availability. You can design one. I have made my own delivery system as well to deliver siRNA. So it is all different, but yeah, choice is yours. Follow the experimental procedure for the viral and non-viral transfection. So after delivery, we need to follow the experimental procedure, like how much we need that cells to be in that environment with the siRNA, because after some time we need to give it a wash also. Even if some of the product will say that you don't need to wash the cells, but I make sure that I remove the excess because it may be toxic to the cell. Then isolate the whole cell RNA after 24 to 72 hours of transfection. So once you transfectate your cells, depends whether you are doing non-viral or viral. I'm talking about non-viral here. Perform the quantitative analysis using qPCR or real-time qPCR. So this is the QR code. Please scan it. You will get the link from the company only, that I made a QR code easy for you, so that you can just scan it and bookmark that particular website. And after the lecture, you can read about the siRNA design criteria. There are different criteria that I have considered while I target my gene sequence. You can do the same thing. So this is one. The second is the Thermo Fisher SiRNA criteria. So what I do is I go through a publication which gives me some criteria, then I compare with the company, because company are making products and they may claim that it works 90% of its efficiency, 90 to 95%. So their chances is higher that whatever they designing siRNA is very potent in their functionality. So I compare this criteria and there is not much difference. I was not able to identify any difference, but the only thing is company are producing siRNA, synthetic siRNA with the modification, whatever you ask for. I have done siRNA with the GFP, fluorophore at the tail, so that once it is inside the cell, I will know how many cells are transfected under the microscope. And that will give me an idea that siRNA is delivered. That is one of the evidence that I can collect, that I have delivered siRNA. If somebody asks, nobody asks because they trust us as a researcher based on the data, but still, some people have that habit of asking, okay, how do I know that your siRNA is being delivered? Okay, I attach the fluorophore so that people can see that, okay, now siRNA is inside the cell. So please go through this PDF. I mean, I think this is the link that I have shared with you. Please bookmark this one as well. This is another. So, there are different levels, but we can convert siRNA to shRNA also if we want to. I have never done personally this, because as a scientist, again, I try to save my time and get the results out quickly and produce more data. So I do not get into the hustle of siRNA to shRNA conversion. But if anybody is interested, if you want to do a small project by yourself, you can convince a PI that, hey, I want to try this one and just see if you can do this. This is a very helpful technique that if you learn it, you can proudly say that, oh, I know how to convert siRNA into shRNA. But these are the few steps that you need to keep in mind. Now, Origin plasmid information. So Origin is one of the commercial company that is here in United States. What we can do is, what I have used there is ShRNA plasmid. I have delivered this ShRNA plasmid without virus and with viruses, both. And they both work. Without viruses, the expression will be transient. But if you want to integrate into the genome, use the viral based shRNA. But you can read about the company, you can know more
Close
about the
your
products.
camera.
Madhubala,
Sorry?
close your camera. I'm talking about Madhubala. Please close your camera. Yes. Please continue, Parth.
Yes. So you can go through this website and learn more about it, learn more about different products. They have give very good sequences available, or everything is available through this website also. I have learned a lot from these companies also, and I have incorporated into my experiment, which has worked. Now, let's talk about the procedure. Now we have siRNA ready or shRNA vector ready. Decide the best transfection reagent for your siRNA or DNA vector, because this is very important, because this transfection reagent has the ability to make small pores in on the cell membrane so that it facilitates the delivery of this siRNA or DNA vector. The follow the procedure to determine the ratio of transfection reagent to siRNA or DNA vector. So when there is a transfection reagent, there is always a ratio coming with the company protocols. How much? 1 is to 2, 2 is to 1, 1 is to 1, 0.5 is to 1. Which one works best for your cells? You need to optimize that by yourself. Once you optimize, you need to use OptiMEM reduce serum media. Why reduce serum media? Because what I have seen that when we use the full serum or full strength, which is 10% of FBS media, the cells, the transfection reagent are not able to make the pores or facilitate the delivery of siRNA or DNA vector inside the cell properly, because they are very healthy. So in order to reduce the integrity of their cell membrane, we need to use OptiMEM reduced serum. That means I have made reduced serum by myself also, which is 1% to max 2.5%. Again, it depends on the cell. I have worked with immune cells, I have worked with human primary cells. There are different cells, so according to the cell type, we have to optimize. And after, once the transfection is done, I change the media after 8 to 12 hours following the transfection, because I don't want extra toxicity and I want cells. So this, what this transfection reagent does is, once they are delivered, 8 to 12 hours of time is good enough for them to deliver the siRNA inside the cell. That's it. I'm not talking about processing. Once they are inside the cell, what I do, I take out this reduced serum media and put the full FBS media so that cells can recover quickly. And then whatever the processing will happen, the protein mRNA will be targeted. That is later on. But this is just the transfection I'm talking about, and I'm teaching you this protocol that I have applied by myself. After changing the media, give 3 washes of PBS to the cells and then add the normal media suitable for cell to grow. And then isolate the RNA after 24 to 48 hours. You need to keep measuring that what is the best activity you see. And so you need to optimize again according to the protein, siRNA, and cell type. And gene knockdown efficiency to target the mRNA or gene using the real-time qPCR or the qPCR. So you can use this. Now, once we transfected, we isolate the mRNA or the whole cell RNA, you can say that. What is the next step? We perform the qPCR or real-time PCR to make sure that we get the transcripts either knocked down, yes or no. So make sure that you have internal control, which is GAPDH, to measure the level of mRNA expression of your gene of interest. So let's say I have X gene I have interest in, I will compare with the gap DH, which is the internal control that has been used by different uh, papers, research that you will see. Must have a gene of interest targeted by the siRNA or shRNA vector to compare it with the normal gene. So now once we determine that how much is that protein or the gene mRNA is expressed at normal level, after the knockdown, we have to compare it again using qPCR. And you will receive the CT values after running a qPCR, real-time qPCR. Then you need to convert CT values to double CT values to 2 raised to minus delta CT values. So this is the step you need to do step-by-step procedure and analysis, because it's a quantitative analysis. It is not qualitative. Because siRNA or shRNA cannot eliminate the protein, is just knockdown. Knockdown is works maximum 80%. So there will be still 20% protein you might be able to see it. But 70 to 80% of the protein has been knocked down. So it has been eliminated from the production. So that's why we use quantitative analysis, not qualitative analysis. And the final value should be plotted after the comparison of these 2 raised to delta CT values between treated and untreated groups. So one with siRNA, one without siRNA or shRNA. The final values of the gene expression should be in the expression fold change unit on the y-axis. I will show you one example that I was able to collect from the published literature. So if you see this is the published literature, and this is one of the figure from this paper that I have put it, which is relative to this hedgehog acylyl transferase catalyzed the sonic hedgehog palmitoylation causes the reduced signaling proliferation and invasiveness of the human carcinoma cell. And they target this HAT enzyme, and you see that the one which is targeted and one is the non-targeting siRNA. People take different siRNA. One is crumbled. You might have heard crumbled or scrambled. Then another is the non-targeting, which is like negative control, because it will not target the gene of your interest. So you will clearly see the difference. Then one people have used internal control. So there are multiple ways which works, but you need to identify according to your application that which one you want to compare, the one with the scrambled one or non-targeting siRNA or targeting some other siRNA. But I think it will not work when you target some other siRNA. So you have to have one siRNA which is positive or experimental, which is yours, that you know that this is the one I want to knock down. The another one that will be, you can compare with the gap DH expression, so that you know that your expressions are changing. And you can check non-targeting or scrambled siRNA. Scrambled siRNA is something which is like it will not have the exact affinity or the binding capacity with gene of your interest that you are targeting. So that way you can compare too. So this is the qPCR analysis example that I wanted to show you, because I have seen students get confused sometimes that which one to use, what is the good way, and sometimes they don't know the right way to present this data. So that's what my today my aim was, to give you this data so that you understand that how you can present and how you can calculate the data after SiRNA or shRNA gene modulation. With that, thank you very much, everybody, for paying attention and listening to this presentation.